Hello, everyone. My name is Marie, and my presentation is going to be on the blood-brain barrier for Human Physiology 1. And I have two objectives in this presentation. One is just to describe the blood-brain barrier and to show details that it is a dynamic mechanism by which the vertebrate brain functions to protect neural tissue. And two, to analyze a study that looks at how the lack of sleep influences the brain through the blood-brain barrier. And here is a graphic of the capillaries of the brain on the right side and the capillaries in the rest of the body on the left side. And on the left side, you can see that normal blood vessels not in the central nervous system actually have openings and are pretty porous, allowing molecules to go in and out. Whereas the brain blood vessels on the right side in the blood-brain barrier actually have tight junctions. And this is an adaptation where it keeps molecules from coming in, blood-borne pathogens, any kind of toxic molecules that may upset the neural tissue on the inside. And this structure ends up being very important for allowing the neural tissue to really be separated from the rest of the body. It was discovered about 125 years ago when they shot blue dye into body cells, and everything turned blue except for the brain and spinal cord. So that was their first clue that this tissue is very, very special. And one of the words that you see sometimes is non-fenestrated. And that simply means that there are no little openings here. It literally means little windows. So the brain blood vessels do not have any openings here. They are very, very tight, as we'll see in a minute. And they are quite different from the normal blood vessels. These endothelial cells are very, very tight together, not allowing anything in or out. And there are efflux transporters and all kinds of transporters that can move certain materials into and out of the central nervous system. The cells of the blood-brain barrier are made up of glial cells in the nervous system, and they help to maintain homeostasis. And three of the main types of cells are endothelial cells, pericytes, and astrocytes. And here you can see that these endothelial cells are very, very tight together. They form a wall. They have many, many mitochondria in them and fewer leukocytes, and they do have adhesion molecules. The pericytes lay along this basal membrane down here, and these are contractile cells that wrap around the capillaries. And then the astrocytes are these purple cells right here, and they actually induce small blood vessels to undergo anatomical and functional changes that establish the blood-brain barrier. So these molecules, they do allow these cells do allow certain molecules to come in and out. They allow oxygen, CO2, steroid hormones, and alcohol to come into the blood-brain barrier and enter into the neural tissue. Water gets in through aquaporins, but otherwise membrane carriers bring in needed materials such as glucose, amino acids, and ions. In fact, the brain uses lots and lots of glucose. In its resting state, the brain uses about 60% of the glucose that is needed by the entire body. So the blood-brain barrier anatomically prevents exchanges of materials, and transport is physiologically prevented or restricted. So it's the entire mechanism that makes up the blood-brain barrier. It acts as a fine sieve. It has advantages and disadvantages. One advantage is that potassium levels do not change. The brain has to maintain homeostasis, especially for the sodium potassium pump that needs to maintain neurons firing off. It prevents blood-borne molecules from entering. It keeps hormones out. There are certain hormones that can interfere and start acting as neurotransmitters. Vasopressin and oxytocin are two peptide hormones that actually are secreted by the pituitary gland, and they actually do function as neurotransmitters. Disadvantages are that it prevents drugs that could possibly help. So scientists spend a lot of time researching and trying to figure out ways to sneak drugs into these gateways into the brain. And there have been some pretty novel discoveries. There's a brain shuttle. There's a technique where they try to pull apart the tight junctions by injecting a hyperosmotic solution. They use ultrasound. Many techniques actually don't work because they lack the target specificity. So there are enzymes that get in the way, and they end up having problems because of that. The paper that I'd like to talk about is, does chronic sleep loss affect the blood-brain barrier? And the aim of this paper was to see if a lack of sleep plays a role in inflammation in the blood-brain barrier. And what does it do with the endothelial cells and epithelial cells? We know that when we have a lack of sleep, there are serious physiological effects from sleep loss: cognitive dysfunction, metabolic alterations, decreased neurogenesis, cardiovascular diseases, immune disturbances. And we all know that when we don't sleep, we really don't feel well. So sleep is really good for us, and it promotes a clearance of these metabolites that load up in our brain during the day. So it is thought that at least 7 hours of sleep is good for us. The authors proposed a hypothetical mechanism by which sleep restriction can induce this disruption by inflammatory molecules. And they used rats in their study. So they took many rats, and the two groups were the control group and the experimental group, where they had chronic sleep restrictions. They actually did distinguish between sleep deprivation, sleep restriction, and sleep fragmentation. And they show in this graphic just one here, and they have different ways of keeping the rats awake. In this particular study, they used this was the platform method, where there is a rod that continuously moves, and they inject them with Evans blue. And Evans blue is an azo dye. It has an affinity for serum albumin, and normal neural tissue will remain unstained. But if the Evans blue is penetrating the brain-blood barrier, then it will end up being blue in the neural tissue. And that's exactly what we see here in the middle diagram, where it says chronic sleep restriction. So this would be the one that actually took up the Evans blue. And then you continue looking down here. This is another side view of brain tissue. And then these diagrams down here are showing blood vessels, and the one that has the sleep restriction actually has more openings. So molecules have penetrated, and inflammatory molecules started to increase in this particular group of rats. And then if we move on here, we can follow the arrows and see that neural damage does occur and cognitive impairment occurs. And I just want to say a word about inflammation. Inflammation, we have to remember, is a defense mechanism. So it's anytime our bodies secrete white blood cells, and it can be triggered by injury or it can be triggered by antigens. So these molecules, in this diagram, you'll see lots of abbreviations here. These are cytokines, and these are molecules that are secreted by the body as an inflammatory response. This diagram shows more of those molecules, and molecules like TNF here is tumor necrosis factor made by monocytes and macrophages. They regulate permeability. The VEGF also is a vascular endothelial growth factor, and the interleukins, which activate coagulation cascades and the release of nitric oxide, all made by these T helper cells. The one pathway here, if we can look on the left side of the diagram, we have an NFB inhibition pathway, and this is one that is found in many parts of the body, actually, and it makes its way as a transcription factor here, and it shows the DNA, and it plays a role in many other metabolic disorders, and this is just one pathway where these molecules are being increased. Over here, the caspase is one of the results, and this will end up causing apoptosis in a cell and causing cell death. So their conclusion was that chronic sleep loss induces systemic low-grade inflammation, and lacking sleep is not good for us. Thank you.